So the membrane potential, this is a difference in charge on each side of the plasma membrane and due to several different factors, including permeability of the membrane, so how many channels are embedded, also the action of the sodium-potassium ATPase pumps that allow for active transport, and then also negatively charged molecules inside the cell. And so difference in charge is what's called a potential difference. So that makes the inside of the cell is more negative. So here we have our cell. So this is a negative cell, and it's more positive on the outside of the cell. There are several ions that are responsible for maintaining a membrane potential. So let's first begin with potassium. Potassium accumulates at high concentrations inside of the cell, and this is due to, number one, the sodium ATPase actively pumping potassium back in. And if you recall, it's 2 potassiums back in. The membrane is also very permeable to potassium. In fact, it's so permeable that the difference is maintained by the potassium concentration gradient. Also, we have negative anions inside the cell attract the positive cations outside of the cell. And it's also limited by strong concentration gradient favoring that for potassium. Another ion that's important in maintaining the membrane potential is sodium. So sodium is going to be less permeable than potassium is, and the membrane potential is closer to that of potassium because of permeability. So what we have in this picture is, here if we just look at potassium, let's just do a little recall. We have a high concentration inside the cell as compared to outside of the cell. However, the reverse is true for sodium, where now we have a higher concentration outside of the cell versus inside the cell. Two other ions that help but are not very important in the process too for membrane potential include chloride, and so chloride is going to be at high concentrations outside of the cell as compared to inside, and then same with calcium, which is going to be high concentrations outside of the cell as compared to inside. The Nernst equation is used to calculate membrane potentials, and so this is based on ion concentrations. And so here we have the equation where EX is going to be the equal potential in millivolts, so that will be our unit, for any ion. So if we're looking at, say, sodium, then we would calculate for sodium and then add potassium and add calcium and add chlorine, all of which contribute to the membrane potential, okay? So XO represents the concentration of ion outside of the cell, Xi represents concentration inside the cell, and Z is the valence ion. So for sodium and potassium, it will be +1. For calcium, it would be +2. And then for chlorine, it would actually be -1. Okay? And then so from that, then the equation is that the equilibrium potential for an ion is equal to 61 divided by that valence ion times the log of the concentration outside of the cell versus inside the cell. And so that will give you for each ion. And then what we do then is add up each ion and how it contributes to the entire membrane potential for a cell. When a cell is at rest, this is what we call the resting membrane potential. And so it does not produce any type of impulse. And that depends on the ratio of the concentrations of each ions on both sides of the membrane, so both inside and outside. And then also the permeability of each ion. So again, the ions that contribute to resting potential are the same. So this is potassium, sodium, calcium, and chlorine. So when a membrane potential of a cell is not producing any impulses, this is due to the fact that the membrane is most permeable to potassium, okay? And so that's going to have the greatest influence in terms of it generating or not generating an impulse. Any change in the permeability of the membrane of an ion, though, can affect the resting potential. And it could be just one ion that travels into the cell and that would offset the entire resting membrane potential. So we're talking about very small changes that can occur in order for it to generate some type of impulse. And so a change in concentration of any of those ions of potassium, sodium, calcium, or chlorine will change then the resting potential. And this is the key to how neurons generate and transmit action potentials. So in most cells, we have a resting potential that's between negative 65 and negative 70 millivolts. Big one that we'll learn about in the next unit is looking at neurons, and so negative 70 is going to be the resting potential. However, within cardiac cells, it will change a little bit and it will actually be closer to negative 65. Okay, so and for all cells though, the resting potential is going to be closer to potassium equilibrium, because for potassium, that equilibrium is equal to negative 90 millivolts, whereas with sodium, that's equal to positive 66 millivolts. So a big difference. So potassium, again, is going to be the determining ion. When we talk about neurons, when they send an impulse, it changes the permeability of sodium, and that drives the membrane potential closer to that of sodium. So that means it's getting farther and farther away from that negative 90 millivolts, and rather it's trying to get closer to that positive 66 millivolts for its membrane potential. So when we look at rest, this is the flow of ions that would like to move. So again, inside the cell, here it's showing it's more negative versus being more positive on the outside of the cell. And so sodium is going to want to come into the cell due to its concentration gradient, whereas potassium wants to leave the cell due to its concentration gradient. If an impulse does occur, then we have activity of that sodium potassium ATPase pump. And so what it does is it helps to counter potassium that leaks out, because there are leak channels that allow for potassium to exit out of the cell. And so again, it transports 2 potassium in and 3 sodium out to maintain a voltage difference. So by this continuous activity, this helps to maintain the resting potential and the concentration differences so that they're stabilized. And so this is considered then to be an electrogenic pump, and for that ATPase sodium or sodium potassium ATPase pump.